Okay, I'm going to talk a little bit about soil health this morning, and I'm going to give you some history of how we've developed this, and then kind of sum that up with how compost plays a major role in this as we go forward. First of all, we have to understand we're in a brand new era of understanding agriculture, and it was coined the evergreen revolution back in about 2011, 2012. And the individuals were looking at the next stage in production agriculture and the reliance on understanding the biological concept of these microbes that help actually the plant feed itself and sustain itself. So we're really looking at understanding soil health. When we look at the definition here, soil health is on everyone's mind today. It's people looking more towards sustainable agriculture with less reliance on fossil fuels and pesticides, and trying to make a more regenerative, sustainable agricultural system. Again, soil health and soil quality are kind of used and kind of overlap at this time, kind of to define that and kind of really define the difference between the two is what I'm going to try to present here today. Basically looking at how these microorganisms help the plant sustain life and help it do a better job in actually producing the crops that we need to provide food for the ever increasing population. So we'll look at the simplified version of the equation of life here, illustrating the central role of soil catalyzation of cycling carbon and nutrients and providing energy to the ecosphere and interfacing the environment with optimizing these nutrient needs for this plant and producibility. So definition of soil health, several definitions of soil health have been proposed during the last few decades. Still a lot of confusion on that, but we're getting to understand it a little more. In the early 90s, it was suggested that soil quality was not limited to soil productivity, but instead expanded to include interactions with the surrounding environment. By the mid 90s, the soil health term was introduced. This was a program to assess and monitor soil health. And at this time, the terms quality and health were used synonymously. And that's the problem even today to some degree. But we're getting to the bottom of that now that we're starting to define what soil health means a little more. And it's broadened to include ecological attributes of soil, which include biodiversity, the food web, structure, and functional measures, including microbial and fungal diversity. So understanding these differences, we still struggle with, and I'll try to elaborate on that a little bit as we go through this presentation. So we'll look at soil quality. Soil quality is the soil's ability to accept and hold and release nutrients and water, promote sustainable root growth, maintain diverse soil life, respond to management practices, and again, resist degradation. So that's where we look at quality. When we look at soil health, it refers to the fitness or ability of a soil to produce high-yielding crops with minimal environmental impact, with reliance on natural soil processes, native microorganisms, and the interactions between plants, animals, and humans, year in and year out. So really kind of drill down to how this whole microcosm of these microbes actually work. And we're getting down to a point where some of these researchers are starting to really define the definition difference between soil quality and soil health. So we look at what we're trying to accomplish here and looking at fundamental healthy soils and understanding this green revolution. It's all about that relationship between plants and their microbiome, the rhizosphere. We look at the rhizosphere of plant similar to our gut. So the rhizosphere is really the gut of the plant, and how this plant actually manages this microbiome of organisms is really important to understand. So where we're at is we are challenged with feeding the population as it grows. We're over 7 billion people on this planet today. We're experiencing decreasing acreage for crop production. So our agricultural land is actually shrinking as we move in, and we're moving into less productive soils. Diets around the world are starting to demand better quality, higher demand for meat and high energy consuming food products. And then there's a big interest and big push to nutrient density in the crops we produce. So there's a real focus on doing a better job of creating better quality food. And again, a lot of this is energy rich food production. So we look at, our approach has always been to look at the quality of the food, the nutritional properties, the nutraceutical components of the crops. And again, we find the soil is the essential link to this whole cycle of life. And how we treat that soil and how we build the soil and its soil health is really important as we move forward in production agriculture. So how do we develop and maintain healthy soils and regenerative microbiome? And what have we learned to date? Well, it's quite astonishing how long this process has taken. We look back to when we actually understood that there was a difference between the bulk soil and the rhizosphere in 1903. Well, Hiltner actually discovered that the plant actually changes the organisms in its rhizosphere. And it's been a long time coming, but it wasn't until about 2011, 2012, we started looking at what is making this change happen. How are these plants changing that rhizosphere, and what are the mechanisms to actually make that happen, and how can we help the plant do that? So again, when we try to understand this microbiome concept and what we have to do to make that whole process work, we have to drill down to the rhizosphere of the plant and just look at what's doing this and how is the plant actually doing that. We do know that 20 to 60% of the photosynthates a plant produces actually end up in the rhizosphere. This is a lot of energy that the plant expends to actually feed these microbes. 15 to 60% of that actually goes to the carbon fraction used by the root to produce and release CO2. So there's an amazing amount of energy that these crops or these plants produce that actually is designed for and used for feeding this microbiome. These compounds, these organic carbon compounds, are all kinds of different organic acids and phenols and amino acids. And this is not by mistake. The plant really controls this chemistry. This is just a simple chart of a book of all kinds of different compounds plants actually produce. When they're giving the resources to do this, they produce these specific compounds to actually sequester these microbes and feed and cultivate these microbes. They cannot do that if we don't give them the proper nutrients and proper environment to actually make that happen. So it's not a simple process. When I started this company back in 1984, I met this individual, Dr. George Lazarzovitz, and he was at that time looking for the disease suppressive soil. He did a lot of work with compost and soil amendments and trying to understand this whole mechanism. He was working globally with other institutions, particularly looking at crop issues like blight in potatoes and scab. So I got involved with him because I actually met him on my partner's farm where he was doing some scab research at that time. For those who don't know George, he worked for the Federal Research Station for many years. He's the guy on the left. So when we looked at this study in Ontario and PEI, two potato producing areas in Ontario that we had research going on, we found out that in Ontario we had less blight, less scab than we did in the Maritime provinces. And we could actually look at the organisms that were different in these soils, and we saw a larger number of what we call the good guys in the Ontario soils than we did in PEI soils. So the question here is, why is this happening? Why are these soils significantly different? At the same time, George was working on a project with Dr. Sean Hemmingson from the NRC here in Saskatchewan, and they were looking at fingerprinting soils from all around the world. They wanted to come up with, if they took a soil sample, could they identify where that soil came from by the microbes that existed in that soil? They were using a new technology of sequencing to come up with these and identify these organisms. They found that there's thousands and hundreds of thousands of organisms they didn't even identify. I mean, you couldn't even figure who they were. So they focused on the top 50,000 or so that they could understand. And their idea here was to say that if we cook a soil from this area versus another, could we actually identify where that soil came from by the population of microbes? What they found out is that was not the case. All soils on the planet contain the same complement of microbes in different compositions, but the same microbes existed everywhere. So we looked at, in our two research areas, Ontario and PEI, and started understanding what that difference was. We analyzed the bulk soil, and you can see here the different microbes we looked at, and the different composition of those microbes in soils. Then we looked at the rhizosphere soil, and again, same microbes in different concentrations. And then we looked at the microbes as they existed as endophytes up inside the plant. And again, we see a little different composition here, but the same microbes occurred. One thing we didn't expect was we actually found rhizobium that we usually would think occupy leguminous plants. We found them in potato roots. So what are they doing there? What are they actually accomplishing? So we're starting to find things out that we didn't expect. But in this case, when we start drill down on rhizobium and the work that's been done today, we know that this particular organism actually stimulates a mechanism for plant defense. So it has a big impact on the plant's ability to ward off disease and infection. More importantly, there's a whole list here of research that's done more recently of disease and insect pressure that rhizobium actually help the plant ward off. And then we also understand that this particular organism produces polysaccharides that actually aggregate soil. So the healthier the soil with a higher concentration of these rhizobium, we get better soil quality, better aggregation of soil particles, better control of water holding capacity and nutrient availability. I mean, the aggregation of these from this rhizobium, you can see here, this is a root hair cross-section of a root of a barley plant, where these polysaccharides are actually gluing the soil particles to that root system to give it better interaction with nutrient uptake. And anybody who's ever pulled a root out of the ground knows how difficult it is to wash soil off of these roots because of this compound. See, these polysaccharides, not only rhizobium, but other organisms produce these compounds. So it has a lot to do with soil aggregation and soil stability. So again, the healthier soil is, and the more of these organisms occur, the more aggregate stability we get. So George told me, or I said to George, how can we take this information to farmers? How are we going to get this out there? And he said, well, the federal government's not going to do this. They don't communicate well at this level. But once the sequencing technology gets to a point where it's affordable, he said, I'll retire from here and we'll start a company. So in 2008, he retired and we started A&L Biological Research Station. And our focus was to direct our whole research on understanding this whole microbiome and these organisms and how the plant actually cultivated these organisms and how that relationship actually happens. In this whole process, we actually looked at what are the microbes doing, how are they there, how do they populate the host, how does the host actually sequester these microbes from the bulk soil into the rhizosphere. So how does that plant signal for these organisms, select for them? How is the host responding to them? So what is the relationship that plant has with these organisms? And what are the unique metabolites or characteristics that these bugs give off to stimulate growth or disease suppression? What are these microbes doing? And what are the forces that maintain the equilibrium among these populations? So really trying to understand how this whole system works. So we look at chemistry, biology, mineralogy. One thing that George told me, because he was a pathologist, he says, my whole career, I looked at sick plants and sick soils. He says, I think to do this project, we have to start looking at highly productive fields and healthy plants to see why they're uniquely different than the plants and soils that are not as productive. So we had the opportunity to meet this individual. He was producing twice the yield of his neighbors in a region, same variety, same cropping systems. And we actually went in his field and we started digging up his plants in his field to actually analyze all these different production zones. George and I, he's a pathologist, I'm a plant physiologist, and we agreed to disagree on a whole bunch of things. And I really told him that I thought that nutrition had a big part of how the plant had this defense mechanism and how it supported this. And he didn't quite agree with that. He said, disease is disease, you can't overcome that. But I said, I think we can suppress it through proper management. First year in, he come to me and he says, Greg, I have noticed something, that the pH of soils has a big impact on how healthy this system is. Someplace in that mid six range, this seems to be our most productive, our highest biological activity. So there is something to this. So we kept looking at that whole concept. When we took the two fields in this particular study, the neighboring field and Dean Glennie's field, and analyzed the difference in what's going on here. In the bulk soil, in early stages, both fields look exactly the same for their biodiversity. But as this field started to progress into 30 and 40 days into emergence, we see a real definite difference in the soil population of microbes. The ones in red, the dots in reds, are the high producing area, and you can see how there's complete difference at about 30 day to 40 day into the production of these two populations. And that maintained itself through through the rhizosphere, up inside the plant as endophytes, right through the growing season, and did not fall back to that diverse population we saw initially until the plant started to senesce. So the plant has completely changed the microbial population, not only in the rhizosphere, but as endophytes within the plant. And, you know, we followed this right through the growth of that plant on a number of different field sites. So we use this technology here called TRFLP, which allows us to isolate communities and really look at the statistical difference between these colonies. So we started looking at the real difference between these populations of organisms in these two areas. When you look at, and one thing we talk about in sustainability and organics is diversity is king. Well, the plant doesn't think that. The plant, in this case, you see the chart on the top, which is an analysis of that microbiome, is quite diverse, as opposed to the one on the bottom, which is a higher producing area. So what has happened here is the plant has actually gone in and eradicated a lot of the freeloaders, a lot of the organisms that are just consuming the compounds it's trying to put back in the rhizosphere from those exudates, so it can have enough food to go around to feed the good guys. So it cleans up the neighborhood and then starts focusing on setting the table for just the ones it wants to keep. So when you look at microbial diversity in the case of a plant and plant growth, it can be very negative. Too many microbes, too much diversity consumes the nutrients, and the plant just can't sustain that population. So in the case of the rhizosphere, the plant is very selective and actually in a healthy soil system can weed out the freeloaders and set the table for the good guys. So in these soils we're working with across these number of fields, we started looking at the chemistry. We looked at the fertility and how it impacted these fields. In the case of Dean Glennie, this is one of the best fields we worked with, and his fertility balance is almost perfect. So we see this relationship now happening to how these balances, in the case K to magnesium ratio, has a big impact on the plant's ability to produce ATP. And these organisms, their whole function in life is to produce ATP. So we see that relationship coming right back to the simple chemistry. And we analyze the good fields compared to the poor fields side by side. The fertility difference in balance was really, really significant different. So we look at the type of respiration we look at. And a lot of people may have used the Savita test kit and looking at microbial respiration. And in a lot of cases, even back when we first started this, we looked at microbial respiration and its connection to soil organic matter. Well, that's not quite the case. This is a case here where we look at Glennie's field with 3.1% organic matter, a CO2 respiration at 67. The neighboring field with lower production at 7.8% organic matter and quite a substantially lower CO2 respiration. And we saw that in a number of cases because we did this CO2 respiration on all the fields we worked with, and it was not always an organic matter relationship. And we'll come back to that a little bit later, but keep that in mind because it's something I think that we focus on, which we kind of miss the mark. So we have a number of fields over the number of years since 2008 that we've worked with. We go in those fields, we send students in the field to collect samples and tissue samples and soil samples and bring them back to the lab. We analyze all the nutrients and the saps of these plants and start looking at what are the organisms we're looking at. So we select those fields with cooperators, again, looking at higher producing areas. We fly the fields and we look at with drones and look at picking out the good and the bad areas and the average areas. And we analyze it statistically across all that with what microbes are in these different areas and what is the actual difference in overall productivity on these fields. We look at yield versus these different nutrients and then compare the endophytes or the microbes in the rhizosphere. And again, we look at pretty much everything. Here you see reactive carbon and organic matter, and you see there's some relationship or correlation to yield in reactive carbon, but not so much organic matter. And again, we'll talk about that a little later. And that's not us. Soliciting all the labs that do Savita tests in the US, they're finding the same thing, that organic matter doesn't necessarily relate to increased CO2 respiration or soil health score. So now we take these different production zones and we look at the microbes that exist in each of these areas. And we start looking at this based on some of the chemistry. In this case, we're looking at nutrition primarily. Anything with a score greater than 0.5 is significant. So look at total plant count here. You can see the general fertility index in the rhizosphere correlates fairly decent. Available potassium. Nitrate nitrogen is not good for microbes. They need some, but too much can actually be very negative to microbial population. And then some of these other nutrients you see, like boron, which surprised me, and then relationship to phosphorus. We'll talk a little bit more here on certain microbes. So look at Pseudomonas, and Pseudomonas is a unique organism. It is a very beneficial organism. It produces all kinds of crazy antibiotics, the right Pseudomonas, because there's differences. It solubilizes phosphate like crazy, but this is the weak sister. It doesn't even come to the party unless things are good. So I need decent fertility, particularly phosphorus, in this soil profile to get this one to work for me. So we see here it has a correlation to increased phosphorus, available phosphorus, but a real magnet for good fertility or good balanced fertility in the soil. So we analyze this particular organism. We look at its response to phosphorus, and we find out that in low phosphate soils, we have more soil-borne diseases such as Fusarium, and certain Pseudomonas actually increase Fusarium load. So we have good and bads in these species. So we have to start looking and pulling them apart to understand how that works and how can we actually define a soil system and a cultural system for producers to do a better job. They solubilize, as I said earlier, they solubilize all kinds of phosphorus and producing antibiotics, but again, not unless they have a decent environment to survive in. We did some more research just on that so we could quantify that a little bit better. This is a chapter we actually wrote in a book of Dr. James White, who's done a lot of work on microbials and their impact on plants, particularly rhizophagy, we'll mention here in a few minutes, but how the plant actually produces root hairs. So we know that with phosphate levels in the 30 to 50 part per million, we have decent Pseudomonas population. We get increase in these organisms with that fertility, but then when we get into phosphate starvation, we start getting opportunistic microbes or microbes that invade the soil space that produce diseases or soil-borne diseases, again, because we don't have that phosphorus that Pseudomonas that's actually secreting these antibiotics. So it's quite something to watch. We also looked, or James White looked at sterilizing soils and getting rid of all microbes to see what kind of an impact it had on nutrient uptake and productivity. And we found out that when that happens, there's virtually no root hairs produced on the plant. So its efficiency to take up nutrients is very much diminished. And then again, diffusion of phosphate in the presence of good bacteria is much more rapid. So when we have good population of bacteria, we get much greater nutrient uptake. So we look at Rhizobium, another very aggressive one I've already talked about, and again, it has a different selection of nutrients that it's good for. So now we know, based on different soil chemistry, which ones we're actually attracting, and we know a way that we can actually stimulate the plant's ability to cultivate these organisms based on having that right nutrient balance. And again, when we look at disease suppression, this particular disease is DON. It's a Fusarium-related disease that is bad for wheat and corn. And we know that these pH, manganese, zinc, boron, percent calcium, and again, things like phosphorus levels have a big impact on the presence of this particular organism. So now we're starting to drill down to how are these different nutrients impacting the plant's ability to protect itself. Another study that we looked at that's not ours, 18-year study looking at fertility levels and balanced fertility, a cue here on balanced fertility. That as we increase balanced fertility, we naturally increase soil organic matter. Without the addition of other organic matter, the biomass returning back is much greater. So we see a significant increase there. We see that balanced fertility significantly increase that biomass that's accumulating, positive influence on microbial populations, and again, particularly Pseudomonas that we really know is one of the key workhorses that we want to maintain. So we have a list of different nutrients that we know that we have control over, that we can actually, once we understand this balance, can actually bring these microbes from the bulk soil into the rhizosphere to help this plant. This is going back to particularly Pseudomonas, but all microbes have an impact on the plant's ability to produce root hair. Without microbes, as I showed you back in that slide, when we sterilized that soil, we had no root hair growth. And it's because without the bacteria that stimulate root hair production in a plant, we do not have an efficient root system. So it has a big impact on how this root system develops and how it takes up nutrients. This is just a simple slide of the same thing, looking at rhizophagy and how this plant cycles these organisms. It takes in the bacteria, it actually strips the cell wall off of these bacteria, it ingests the nutrients that's in it, and it consumes those bacteria, believe it or not, as a protein source and nitrogen source. So those vegetarians in a room, plants are meat eaters. Okay, so just keep that in mind. So nutrient availability determines endophytic survival. Endophytes are very specific to the carbon source they want, or the nutrients that the plant gives off to support them. Competition for those nutrients can be a very determining factor in how this whole community is shaped. So again, understanding that balance allows a producer to really work on improving his soil health through cultural practice he can actually do. And then specific nutrient availability may limit the number of endophytic microbes that can be taken up and survive inside the plant tissue. So there's a bunch of things that we can do, and it gets down to, what have we learned from all this, and how can we help build a healthy soil? So what makes up a healthy soil? Ideal pH is number one, because again, right from the day one, when George and I identified that, that pH around 6.5 seems to be the happiest environment for both fungi and bacteria, it's really important to focus on that whole concept. Balanced nutrient availability, and emphasize balance. The problem we have in production agriculture in North America is when we talk about fertilizer, everybody's focused on nitrogen. Nitrogen is detrimental to some of these. We need some, but it can be detrimental if it's abused or overused. It's the other nutrients that are more important in sustaining this population. Good moisture holding capacity. Number one, aerobic soil conditions, no compaction, aggregate stability, big deal. A stable fine fraction pool of soil organic matter with good CNPS ratios. So that relationship between carbon and nitrogen ratio is really important in the organics that we put on these soils. So we look at soil health. It's a lot about the chemistry, and we can actually have some impact on that. We built a soil test that has a soil health index on it using chemistry and looking at, when I look at this chart, I can tell you who's in the rhizosphere based on some of the chemistry balance that we have here. Is it heavy in Pseudomonas? Is it heavy in Rhizobium? Is it heavy in Bacillus? It's all in that research that we've put together in understanding the soil health concept. The soil health algorithm actually has 487 different factors that we've used to build that. So it's very complex, but there's things there that we can educate farmers on how to amend their soils to do a better job in building this healthy microbiome for crop production. The important factor here is we got to understand that soil moisture plays a key role. Microbes need moisture to perform. They need this happy environment to sustain their ability to help the plant. In 2015, one of our better years, we had a 93% correlation to this soil health algorithm we developed. But over the next years, we saw that change quite a bit. And it was all based on soil moisture. 2016, we had a real drop in the relationship to this algorithm we produced because we had less microbial activity, because we just had a drought in those areas and just no soil moisture. So that's important when we talk about compost as an amending product for soil and soil health. If you do a good job composting, which you all do, I'm sure, and you look at our soil health test, our not a soil health test, but our S8 in the CQA, this gives you a good understanding of the compost that you're building. Does it have the right nutrient balance to sustain a healthy soil profile? We're looking at a compost here, which I call a good compost. It has the right ratios and nutrients. It has very good phosphate load in it. It has high nutrient load in general. Not that I want to plant a plant in this, because it's probably too hot for these nutrients that would likely cause some problems. But if I take this compost and mend soils with it, at even as much as 1,000 pounds to the acre, I'm going to retain more soil moisture. I'm going to have slowly available nutrients that's in a good balance, because the pH is where it should be, that I'm going to have a good nutrient source for this microbiome to feed these organisms. So I'm helping to promote and sustain a very a very lush environment for these microbes, and particularly of notice here, the ability for soil moisture. And when I showed you those slides earlier about organic matter having not a big impact on yield, it was primarily because in dry weather conditions, we see it, but in conditions where we have all kinds of moisture, it wasn't quite as statistically significant. So where organic matter plays a role is creating that nice, happy environment for these microbes, and particularly the moisture retention is critical for every living organism on this planet. So when you look at adding organic materials to soil structure, number one, we want an aerobic condition. Huge on water holding capacity. Like I said, 1,000 pounds to the acre has a big impact on moisture retention in growing seasons. Drainage, infiltration, aeration, nutrient cycling. Again, that slow release of good available balanced nutrition, not just N, P, and K, but all the micronutrients that are just as essential. And then that whole soil biological activity that's so important. So you look at compost and what it can do. What does increase in organic matter do? 0.5% increase in organic matter increases 15% in nutrient holding capacity, and then 12% in water holding capacity. Just 0.5% increase in organic material. So as you improve your soil, you improve your crop improves, and we can actually see an increase in water holding capacity with very small amounts amended to soils to increase yields. So just kind of some calculations and factors here. We look at how important that is. 1.5% to 2% organic matter can hold 35 to 45 pounds of water, equal to 0.5 to 1.5 inches of rainfall. 4 to 5% organic matter can hold 165 to 195 pounds of water, equal to 4 to 6% of rain. So we really see how important this is. And water, everybody understands how essential water is to life. And, how can we maintain that in droughty soil conditions? And as we get moved off of prime agricultural land into land that's not nearly as productive, one of the basic limiting factors is organic matter. And we just don't have enough of it to retain the moisture and produce or maintain this happy environment for that microbial interaction with plants and soil health. So again, I did a research work back in the 90s in southwestern Ontario, and we established back then if we took everything we could out of the waste stream that we could compost and composted it, we would have enough organics to satisfy the needs of 2 counties. So we have a real need for these organics, and we need to understand how we need to produce them. We need to understand what is a good compost and what is a well, there's no such thing as bad compost. Compost has a place, someplace, but when we look at putting it on agricultural land for agricultural use, we can actually be detrimental in some of the compost we produce that might be too high in sodium or might be low pH. So we need to know when we're putting compost together what it is actually good for, and use it in that regard. And don't try to put a compost that has some of these less than ideal qualities into agricultural production, because you could do more harm than good. But for the most part, we just need more organics. So with that, I thank you and hope that kind of helped you understand soil health.